Hello.
So second speaker is Kinat Keren from the Technion Institute in Israel. She will talk about physical aspects of hydra morphogenesis. And thank you, Kineret, for being with us.
So thank you, and it's unfortunate that I cannot be with you there in Paris, so I'll talk to my computer screen from here. I'm going to talk about our work on hydra morphogenesis, and you're welcome to interrupt me with questions during my presentation. So my lab is interested in mechanical aspects of biological self-organization and pattern formation, and we've worked much at the single cell level, looking at actin-based cell motility and on the cytoskeletal system encapsulated in artificial cells. But today I'll tell you about our work on morphogenesis on the supracellular scale using hydra as a model system. So morphogenesis is one of the most beautiful examples of pattern formation in nature. As you can see here in these images of a human embryo developing, you have elaborate functional forms developing over time, and it's really a fascinating example of pattern formation. And one of the most striking features of this process is how robust it is. So what's called canalization, you look at the process of morphogenesis, and the outcome is extremely persistent and consistent, even though there is a lot of variation, both internally in the system, in its genetics and in its properties, as well as in the environment. Yet when you look, for example, on a human embryo develop, you almost invariably get a baby with 2 hands, 2 eyes, a nose, this is really striking. And one of the central questions in this field is to understand how robust patterns emerge in morphogenesis. And just to give 2 examples that exemplify this remarkable robustness, here I've just taken 2 examples of features that are obviously important for the development. One is temperature. So temperature will influence all the underlying processes involved in morphogenesis. Yet when you look at, for example, a fly develop, it can develop over a spectrum of temperature. And temperature obviously matters because the time it takes to develop will vary over 7-fold when you change the temperature from 12 degrees to 28 degrees. Yet you will end up with a fly with the same pattern. Similarly with size, if you take eggs of different sizes, you'll get a small chick. You also know that from humans, you can take eggs, split it in half, and you'll get identical twins. So the developing organisms are able to cope with a lot of variation and develop robust patterns. And it's really a striking and intriguing question how this happens. Another example of this process of morphogenesis is what's known as regeneration. If you take a mature organism and it undergoes some damage or some injury, it's able in many cases to recover. So for example, if you take a salamandra and you chop off its limb, a new limb will grow within a few weeks. And this is a complicated tissue. It contains skins and muscles and nerves and bones, and all this has to grow into a functional limb, and it's able to recover. In humans, we're not as advanced in regeneration, but you know that if, for example, a bone breaks, it's able to regrow, or a liver can regrow. And in some animals, like planaria and the hydra that I will be talking about to you today, the system is able to undergo what's called full body regeneration. So if you take, for example, a planaria and you chop off its tail, the planaria will grow a new tail, but also the tail will grow a new animal. So in these systems, you're able to take a part of the system and regrow the entire organism. And if you think about this process of regeneration in analogy to development, you end up with the same form, with a mature animal. In development, you start with a fertilized egg and you go along this well-defined trajectory of embryonic development. In regeneration, you actually start from arbitrary initial conditions. So wherever whatever damage happens, you have to deal with these initial conditions and accommodate existing parts, yet generate this very well-defined functional adult pattern. So in terms of the robustness, this is another very nice example of this, where you start from essentially arbitrary conditions, yet you're able to generate the same well-defined patterns. And in fact, looking into the underlying mechanism, regeneration and development actually share both on the molecular side as well as on the physical process. There are shared mechanisms that are responsible for both of these processes. So in general, if we're thinking of morphogenesis, there are two main approaches to this problem, and they're actually complementary. On the one hand, we have what's called the program view. In this view, you can think of development as kind of an assembly line, like a car assembly line. So you think of a pattern that unfolds along a predetermined route, step by step, you form this elaborate pattern. So on the one hand, when you look at the developmental trajectory, this very much looks like an assembly line in the sense that you see the pattern forming step after step in a very stereotypic manner. However, it's really not clear how robustness would emerge in such a step-by-step program. For example, if your car breaks down in an accident, you don't expect to go back to the assembly line and fix it, but you'll go to a car mechanic. On the other hand, you have the more dynamic view, the self-organization view, where you think of the pattern developing through a dynamical process rather than a predetermined route. This takes its analogy from the field of active matter, and you can see an example here with this flock of birds, which develops into these patterns through the dynamics of the process. And if I take this flock of birds and somehow, for example, it flies across a tree and it gets split, you can understand how it will dynamically regroup, and you can understand how feedbacks will play into the pattern, and perhaps you can understand how these patterns can be robust. But in fact, if we think of self-organization, we have no examples that are near as complex as what we see in morphogenesis. So if we want to look at the underlying mechanisms responsible for morphogenesis, the main view kind of emphasizes genetic networks, signaling molecules. There's been a lot of work over the last few decades in identifying a lot of the molecular components which are involved in intricate networks of interactions that eventually are thought to lead to patterns. And one of the systems where this has been most studied is the fly embryo. So here you can see examples of images of developing fly embryo taken from Eric Wischhaus, who got a Nobel Prize on his contributions to the genetic control of early embryonic development in the flies. So what you're seeing here are patterns of molecules. So each step, in the first step, you have some pattern that is actually initialized from the mother of the embryo. And over time, this develops into a gradient in the protein concentration and different types of molecules generating more and more elaborate patterns. And the general framework for thinking about how these concentration differences translates into patterns are the idea of reaction diffusion processes. So we have this concentration of molecules describing concentration of these important signaling molecules that generate the pattern, which are called morphogenes. So these morphogenes their concentration will vary in space and time, and they can react and diffuse, and there can be other means of transport. And this, as shown by Turing in his seminal work, can lead to patterns. And then these patterns of concentration gradients are translated into patterns in the developing embryo through positional information encoded in these concentration gradients, which is translated into patterns in the cells. So the basic idea is that there are these biochemical morphogens, these signaling molecules that generate these spatial concentration gradients with more and more elaborate patterning that translate into the pattern that we see in the cells. So this focuses on the biochemical side of the patterning process, but in fact, it's becoming more and more appreciated that mechanics plays an active role in the patterning. So not only follows the directions instilled by these genetic networks, but actually is an active part of the patterning process. So ultimately, morphogenesis is obviously mechanical processes. The tissue folds and deforms. So these are all mechanical processes. And one of the first people who considered that the mechanics can, that the physical processes and the mechanical forces involved in this can actually help us gain insight into the patterning process itself was Darcy Thompson, who realized that these various constraints and thinking about from a mathematical and physical point of view on these patterning process can lead to a lot of insight into which patterns can actually form. In the last few decades, there's been a lot of work looking at how mechanical skews can in fact also interact and drive patterns and inform biochemical processes at all levels of organization, from the molecular through the cellular to the tissue scale, where the basic idea is that there are various processes where mechanical cues, mechanical signals, for example, the stress and strain in the tissue, can feed into the behavior of molecules or cells or even at the tissue scale. And these feedbacks are actually very important because they allow you to connect the pattern that emerges with the underlying processes that are building it. And you can see an example of this here. So if we consider a growing tissue, then there will be a stress and a strain pattern developing in this tissue. If, for example, cells are able to respond to these stresses and change their behavior, this will in turn change the dynamics of the tissue and change its behavior, feeding back into the stress and strain patterns. So this allows you to connect the pattern that emerges with the process that is shaping this pattern. And this is becoming more and more, it's becoming more and more appreciated that this is an essential part of the patterning process. So we have the biochemical and genetic networks that are generating patterns, but in parallel, we have all these mechanical processes that are also actively shaping the pattern that emerge. So when we think of this process of morphogenesis, we really need to think of it as an integrated process where we have the biochemical, the signaling pathways, the genetic interaction, we have mechanical processes, and there's continuous feedback between these two types of processes. And this feedback spans all the scales, from the molecular level through the changes in cell behavior, depending on the mechanical cues that they see, to the tissue level. So from a physical point of view, we have here a very complicated, many bodies, strongly interaction, far from equilibrium dynamical system. And what's striking is that it's able to self-organize across all these scales to generate robust patterns. So when we think of this self-organization process across scales, as I mentioned, we have the molecular level, which is important, where we have biosignaling networks, cytoskeletal dynamics. We have the cellular level, where cells can move, adhere to each other, differentiate, divide, die. We have the tissue levels, where we see the tissue forming, stress patterns, deformations occurring all the way to the whole organism scale. And all these are feedbacks between all levels of organization, and there's no clear timescale separation or spatial scale separation. So it's really a complicated multi-scale dynamical system. And a lot of the research in this field focuses on a reductionist approach, where you seek to identify the molecular players involved, which molecules are, identify the morphogen, identify their interactions, and try to build bottom-up models to understand this process. But we know from physics that when we have complex multi-scale dynamical systems, it's actually also very useful to take an effective approach where you look at the system kind of from afar a bit. You look at a mesoscale description where you don't focus on all the molecular details, but try to identify some coarse-grained variables, some variables that are on a larger scale, not the detailed molecular structure, but on a more coarse-grained scale, and try to develop effective models for the dynamics. And these can be extremely useful. Obviously, a lot of molecular details are not taken into account in this approach, yet it enables us to bridge all the way to the macroscopic and whole organism approach and obtain predictive descriptions that can be very valuable. So this is the approach we've been taking to this problem of morphogenesis. And what I want to do today is first introduce you to Hydra, which is the model system we've been using in our work, and explain to you why I think this is a great model system for this problem, as it is, on the one hand, rich enough to address this problem of how pattern emerges in a multicellular animal, yet on the other hand, relatively simple and flexible experimental platform to look at this problem. And then I'll talk in the first part of my talk, I'll present our work looking at the supracellular actin fiber organization in regenerating Hydra. So we're looking at the organization of supracellular actin fibers. So they vary in cells that are on scales that are supracellular, much larger than an individual cells. And what we see is that the organization of these fibers, and in particular defects in the organization of these fibers, actually act as organization centers in the process of morphogenesis in these regenerating Hydra. In the second part of my talk, I'll focus on polarity in regenerating Hydra. I'll show you experiments where we've developed a way to introduce frustration. So typically, there is a very strong memory of polarity during regeneration. If you take a piece of tissue, it will remember its original polarity and regenerate accordingly. But what we've done is generated conditions where polarity cannot be inherited simply. We introduce frustration by generating ring doublets with opposing polarities. And by looking at how the system can respond to this frustration, we can learn about the processes involved in polarity determination. And in both of these parts, what will come up very nicely is that the mechanics in these, the mechanical field associated with the organization of the supracellular actin fibers, this coarse-grained mechanical field is very important for the regeneration process and for determining the axis of the animal, but it's intimately coupled to signaling processes associated with the patterning, and it's really this interplay that leads to the robust regeneration of Hydra. And to summarize and conclude, I want to put forward this idea that in analogy to the biochemical morphogens that we are familiar with from as the drivers of patterning in developmental, we can also think of mechanical morphogens, which again are not acting on alone, but interact with these biochemical morphogens and together lead to the patterns that we see. Okay, so after a brief introduction, let me introduce you to the start of today's talk, which is Hydra. So this is a small multicellular predatory animal. It lives in fresh waters and in ponds and etc. It's if you look at the animal tree of life, you can see that it's part of the Cnidarian family. This is the same family as jellyfish and coral that diverged very early in the animal kingdom. It has a very simple body plan. It basically has a single axis with a foot on one side and a head on the other side. On the head, there's a mouth at the tip of the head, and then there are these tentacles which it uses to catch its prey. It has about 100,000 cell types. It has about 100,000 cells, about 10 different cell types. It actually has a distributed primitive neural system. It's actually also being used as a nice model system where you can look at the entire neuronal system. And it gets its name from the mythological Hydra. So the mythological Hydra, if you cut its head, it would regrow a head. And Hydra, which was first actually studied by Abraham Tremblay in the 18th century, is able to regenerate if you chop it into parts. So if you cut it into half, each half will grow the missing half. You can cut it into many pieces, and each piece will develop into a Hydra, as I'll show you today. And you can even put it in the blender, dissociate it completely into individual cells, reaggregate them, and again, regenerate a complete animal. So when thinking of the process of morphogenesis, of the development of adult animal, we see that the Hydra gives us a very elaborate flexibility in the initial conditions for regeneration. This process of regeneration does not require growth, actually. So if you can stop cell division altogether and you will still get regeneration. So it's basically the same cells that can reorganize and differentiate to generate the form of the adult animal. Regeneration happens in solution, which means that it's totally accessible for us in the lab. And the entire process is quick, so you can go from start to finish within a couple of days, which again, from an experimental point of view, is a great asset. Okay, so this is how this process of regeneration looks. So what you're seeing here is a movie where we took a hydra, we took a small piece, about 5% of the whole animal from the middle part of the animal. This piece of tissue, the first thing it does is it folds into a holospheroid. And what you can see here in the movie is the process of how it regenerates over the next couple of days. So the time here is in hours. You can see the tissue dynamics. You can see these osmotic inflations and ruptures. You can see by now, you can see a clear axis forming. You can see the little tentacles forming, and you see the new head and the new foot. And we have a regenerated animal. And all this took less than 2 days. Normally, if you look at hydra, it reproduces by budding. So the typical way hydra reproduces, a small bud starts growing from the mature animal and then dissociate. If you put it under stress, it's also capable of sexual reproduction. So it will produce oocytes and testes and undergo sexual reproduction. And in general, if you look at an adult hydra, it's actually in a continuous state of development. So there's continuously cell division and movement towards the extremities. And in fact, hydra have been shown not to age. So basically, they live on forever, replacing all their cells. But the organism itself, as far as we know, can live forever. Okay. A little bit more about the structure of the hydra. So as I said, it basically has a single main axis from head to foot. So it's basically a tube. So it's a hollow tube made of a bilayer of cells. So you can see here in a cross section. So we have this tubular structure with the axis from head to foot. And the animal body is actually made of these 2 layers of epithelial cells. Here you can see in a cross section, the outer layer is called the ectoderm. The inner layer is called the endoderm. In between, there's a thin matrix of cells. And these cells, they're actually double as epithelial and muscular cells. So they're both the kind of outer and inner skin of the hydra, but they also contain muscle fibers. These muscle fibers are on the basal side of both of these epithelial layer, and they're oriented in perpendicular direction. So if you look at the outer ectoderm, you can see this beautiful array of supracellular fibers going all the way across the entire animal. So you can see here the scale of individual cells, and you can appreciate that these are really supracellular fibers that can mechanically connect across millimeter scales. In the inner layer, you have a perpendicular layer of supracellular muscle fibers. So here you see these fibers in a small hydra, and you can see that these are circumferential along the body. So why is hydra such a nice model system for us? So on the one hand, it's a multicellular behaving animal. So you can see here in this movie, this is a movie from YouTube of a hydra feeding on small shrimps. So it paralyzes it with its tentacles, brings it to its mouth. It will open its mouth and digest this little shrimp, which is about the same size as the hydra, and then digest it for a few hours and spit the remnants through its mouth. So on the one hand, we have here a multicellular behaving animal. On the other hand, it's rather simple. It has a simple body plan. It has these remarkable regeneration capabilities that I showed you. From an experimental point of view, it's really a flexible model system because of that. We have this variable initial conditions for morphogenesis. It's very amenable to manipulation because everything is happening in solution, and it's easy to work with. And we can also have, as you see, transgenic animals. So it's really complicated enough to ask the question, yet relatively simple to be a more tractable model system. Okay, so after this kind of short introduction to familiarize you with hydra, let me go on to the first part of my talk, where we're focusing on the supracellular actin fibers and looking at how its dynamic organization during regeneration. So as I've already showed you, if we look at a mature hydra, we see this beautiful parallel array of supracellular fibers that spans the entire body of the animal. Here you can see a zoom in on these fibers, and this type of organization, where you have parallel objects which are aligned along a preferred orientation, is what's called a nematic orientation. And we can describe this organization by associating a director field. So basically a headless vector that just points to the average direction in each region. Computationally, we actually use software developed for fingerprint analysis to look at image intensity gradients and extract the direction of these actin fibers and get these what's called director fields. So basically a field that at each point gives us the direction along which the fibers are oriented. Just to show you that this orientation field is well defined in the animal, if we look at here, zoom in on a part of the tissue. What you're seeing here, just images from a time-lapse movie taken 1 minute with 1 minute time interval of the epithelium. And in this case, we caught a cell that is dividing. So you can see here a cell, it's rounding up and actually dividing into 2 cells. And you can see throughout this process that the actin fibers actually remained intact. So we have this supracellular orientation field defined by the orientation of these supercellular fibers, and it's well defined over scales that are much larger than the kind of molecular scale or even cellular scales, and processes like cell division do not alter this orientation field. Okay, an important part of the organization of the nematic organization is the presence of defects. So I've told you that the nematic material tends to orient parallel to itself everywhere, but there can be these defects or points of singularities in the field, as you can see here. So you can see that the actin fibers are parallel everywhere, but there is a point of singularity or defect in this place. And we can actually characterize these defects according to and categorize them into different defect types and associate a charge to these defects. And this charge is basically just the winding number. So if I go around a defect, so I have a defect here. If I go on a path around the defect and return to the same place, the director field has rotated. And in this case, it rotated a full rotation. So we have a defect of +1. If we take a defect of +½ it would have rotated a half direction, and here -½ means that it rotated a half in the other direction. And these charges are actually constrained by the topology of the system. So if we have a system like a hydra, which is on a holospheroid, the total charge of all the defects in the system has to be +2. And this is just a mathematical statement on these charges. Okay, so getting back to the hydra, if we look in a mature hydra, we see that there are, so we have this nematic alignment in most of the animal, but there are also defects, and we see that these defects are not randomly distributed, but they're actually localized at the morphological features of the animal. So we have a +1 defect at the tip of the mouth, we have defects at the tip of the foot of the animal, and then we have +1 defect at the tip of each tentacle and -½ defect at the base of each tentacle. As I said, if we sum up all the charges, we have to get to 2, and this is indeed what happens. So we have the head and the foot, each +1 gives us +2. And then we have a varying number of tentacles, but they actually don't contribute to the total charge because the +1 defect at the tip of the tentacle is balanced by the 2 -½ at the base. And in cases where we have, for example, hydra with multi-heads, so if you have a two-headed hydra, as you can see in this image, we'll have an extra +1 defect in the extra head, but this will be balanced by 2 -½ defects at the junctions. Okay, so this is the organization of the actin fibers in mature hydra. We have this nematic alignment of the supracellular actin fibers. All the fibers I've shown you, these are in the ectoderm of the animal, and they form basically a two-dimensional surface on the basal side of the ectoderm. And in the adult hydra, they're arranged with defects associated with coinciding with the morphological features of the animal. If we look at the endoderm, so the inner surface where I said there are perpendicular wires, we see the same pattern of defects. They have the +1 defect in the head and mouth and in the tentacle. In this case, because the fibers are oriented circumferentially, we have, rather than the aster type +1 vortices, we have the vortex type +1 vortices. So now, okay, we understand how the actin fibers are organized in a mature hydra. Let's look at the process of regeneration. So when we look at regeneration, what we do is we cut a piece of tissue from the animal. So if we come with a scalpel and you cut a piece of tissue from the body of the hydra, it will actually inherit these supracellular fibers. So what we've shown already a few years back is that this inherited organization of the cytoskeleton actually confirms a structural memory and actually directs the body axis of the regenerated animal. So we have this direction which is associated with the orientation of the actin fibers, and these fibers will provide a memory of the original body axis. Okay, how does that happen? Actually, it's not trivial because of the folding process. So if we take a piece of tissue, so I take a square piece of tissue, and the first thing that it does is it folds into a spheroid. And when it folds into a spheroid, it cannot maintain this alignment everywhere. And indeed, what we see is that when we look at the folded spheroid, and this process is rather quick, within about 2 hours, we'll have a sealed spheroid. What we see is that the actin fibers in the central part of the fragment are actually maintained, but in the closure region, the fiber are lost. So what you're seeing here is a spheroid where we're looking at the same spheroid from front and back. So two opposite orientations. The spheroid actually folds. If you see me on the camera, it actually folds. So it folds and kind of into a closed spheroid. The part that was in the center of the fragment is the part where the orientation of the fibers remain. The part in the closure region is the half where we see that the fibers are lost. Okay, so the first thing that we see happening over time is after this initial folding, the spheroid starts to regenerate. And what we see is that there is induction of order. So if we look at the fraction of the area that contains ordered fibers, initially in the folded spheroid, it's about half. So what you're seeing here, this is the ordered area fraction. So the fraction of the area of the spheroid that contains this parallel alignment of fibers, whereas this region we consider disordered. And you're seeing it over time over regeneration time, where each line here is what we've extracted from individual light sheet movies, where we're looking at the dynamics of regeneration from all directions. So what you see is that we start off with about half of the tissue with ordered fibers, and over time this increases until it saturates and remains at a value that's close to 1, except the regions close to the defect. Okay, so we have the system. Initially it has no defect, perfect alignment, but it folds, and then there's a large disordered region. But it also changed the topology, and therefore defects must arise. And the types of defects that we see in regenerating hydra can be characterized into this +half, -half, or +1 defects, as you can see here. So on the top, you see the fluorescent images of the actin fibers. On the bottom, what you're seeing is a zoomed region where you see the local alignment. So the bars show you the local alignment of the actin fiber extracted computationally from the images. And then the color basically gives you the degree of order, what's called the local order parameter. This is just averaging in a region the alignment, the local alignment of the fibers. So in regions where all the fibers are aligned, this just gives you 1. So this will be colored red. In regions close to the defect, this will be smaller because you have fibers from all orientations. So this average of the cosine of the relative angle will be smaller, and you get minima of the local order parameter. This is very useful because it allows you visually to pinpoint where the defects are. So we have these +half, -half, and +1 defects in the animal. And when we're looking, so we're looking at this process of morphogenesis. The tissue folds. We know it has to. This has to introduce defects. We see that these defects are long-lived. If you look at the whole animal, I've shown you earlier that the defects coincide with the morphological features in the animal. And the question is, can we learn from looking at this process of regeneration from the viewpoint of the actin fiber organization, get an effective picture of this morphogenesis process by looking at the defect dynamics, because we have these relatively stable defects. At the end of the day, we know that the defects should localize to the morphological features. Can we understand something about this process by focusing on the actin fiber organization and the defects? Okay, so this is a movie showing how these dynamics look like. So what you're seeing here, this is a tissue, and you're seeing it from 4 different orientations. So this is basically a light sheet movie. On the top, you see the fluorescence of the life pack showing you the actin fiber organization. On the bottom, you see the local orientation field and the local order parameters. So the color gives you a sense of the defects. You can see initially we have this, half of the spheroid has nice fibers organized. So this is this region here. There are other regions which are disordered. In the local order parameter, these are highlighted. So you can see these highlighted region. And if I play the movie, you can see the dynamics of the tissue over time. Again, the tissue is very flexible, deforms, changes shape. There are osmotic inflations and ruptures as well. You can see the induction of order. So by now you see that most of the regions are already have the nematic organization. You can see the formation of defects. So you can see these regions that form, and you can see that they're, the tissue is deforming, but that these defects are rather stable. And you can start seeing the pattern develop. So you see the tentacles emerging, the mouth forming, and the foot. So if we look at the final images, we can see the defects. We can clearly see the +1 defect at the tip of the mouth. We can see the tentacles forming. We can see the +1 defect at the foot. So we can track these the dynamics of the supracellular actin fiber organization through time and follow how the defect pattern emerges and changes as the animal develops. Okay, so what do we see? So if we look at the, so initially we have no localized defects. Initially we're looking at the flat piece of tissue with an open boundary. There are no defects. It's perfectly aligned and it has open boundary. After a few hours, it's already sealed. We don't have defects. What we have is a large disordered region. Over the first 24 hours, as I showed you earlier, there's an induction of order. So all the regions that are disordered become ordered, and we have nematic order emerge everywhere. But this means that we now have to have localized defects. And when we look at which defects are forming, so what you're seeing here is the defects that we counted in the average defect number that we counted in different samples over time. And what you see that initially, what we see is that +½ defects are the most common form of defect. So you see that +½ defects form, and we see a large number of +½ defects appearing at 24 hours. However, +½ defects are totally absent from the developed animal, and indeed we see them disappear over time. If we're focusing on the defects only in the body, ignoring the tentacles, there are hardly any -½ defects. And the +1 defects that eventually will be present in the mouth and the foot develop over time, as you can see here. If we look at the total charge that we see here, is initially in this period where we still don't have order everywhere, the total charge of defect is not 2 because we have large disordered regions. But once we have a nematic order everywhere, the total charge is constrained to be 2, and then it remains 2, as you can see in this graph, which basically we're plotting the net charge of all the defects found as a function of time. Okay, so let's look a little bit more closer on the dynamics of the defects. So if we look at the +½ defects, these are asymmetric in their structure. And what we see is that they're moving into the tissue. Now, experimentally, this was quite a challenge to look at the dynamics of the defects relative to the tissue, because as you saw, the tissue is deforming and changing its shape a lot, and it's very hard to track the tissue. So to be able to mark specific points in the tissue, what we did is develop a way to introduce a caged fluorescent dye everywhere, and then use a laser to uncage it in particular regions. And using this approach, we were able to label particular bunch of cells in this region. You can see a +½ defect here. And then we can follow this region of cells. So this label goes into the cells and is stably remains in these cells. So it allows us to track these cells over time. And we can follow them relative to the defect. And what you can see is that the defect is actually moving relative to the tissue. So you see the defect here was closer, and then over time moves away from these cells. And the direction of the movement of the defect is, as I plotted here, away from its rounded edge. Okay, so the +½ defect are actually mobile in the system. I told you before that they appear, but then they disappear. So how do they disappear? The most common way that they disappear is that we see merging of two half defects, and this is typically accompanied by elongation of the tissue, and eventually they merge to form a +1 defect. Another way they can disappear, which is more rare, is by annihilation. So if you have a +½ defect and a -½ defect, and they're oriented so the +½ defect moving towards the -½ defect, what we see is that these can annihilate each other and we get rid of the defect. So these +½ defects disappear mostly by merging, but sometimes by annihilation. What happens with the, what was particularly striking is that this formation of these defects was actually directly associated with the morphological processes happening in the tissue. So the main things that are happening in the tissue is the formation of the head and the formation of the foot. When we look at the merging of these two +½ defects, so you see the two +½ defects, eventually they will merge. This will typically become the foot of the animal. So you can see here, when we look at these, when we identify regions of two +½ defects merging and forming one, this will typically end up being the foot of the animal. And when we identify +1 defects early on in the regeneration process, these +1 defects remain stationary, so they're not moving relative to the underlying tissue, and eventually will end up being the head of the regenerated animal. So what this implies is that we can, just by looking at the nematic field, we can identify the regions that will become the head or the foot of the animal just from looking at the organization of the supracellular actin fibers and identifying these defects, which appear rather early, way before, as you can see here, way before we see any morphological features appearing in the tissue. Okay, so what this implies is that actually we can identify the regions where the defect are occurring as the organization centers of the morphogenesis process. So I showed you I can identify defects. They remain stable over time. So if I look at the cells that are located in these defects, these are still localized in the defect region throughout the regeneration process. If I think of these defect sites, these will have a unique mechanical environment, and this just follows from the definition of these defects as the center of these, this aster organization of these contractile fibers around the defect site. So these supracellular actin fibers, they're part of the cell-cell adhesion between cells, and they're contractile. So when they contract, they will generate a force. So if I look at the defect region will be characterized by having no fibers crossing it. So there will be weaker adhesion typically in this region. And also, if all these contractile fibers decide to contract, there will be high tension in this region. And indeed, I told you earlier that the tissue undergoes osmotic inflations and ruptures. You saw in the movies that there's these inflations and ruptures. The release of osmotic pressure is done through ruptures in the tissue, and holes are forming. And we see that these holes don't form randomly in the tissue. Once we have the +1 defect, these holes invariably form where we have at the defect sites. And at the end of the process, I told you that these +1 defect sites will become the head of the animal. So what this means is that we can identify, by looking at the defect, we can identify sites early on in the regeneration process, and we know that these sites will be the sites where the morphological feature will appear. So if you look at early +1 defects, we can identify this as the region where the new organizer of the hydra will form and the new head of the animal will appear. Question still here?
Yeah, so I have a question.
Yeah.
Can you go back? There was the other graph with the two half defects that generate the foot.
Here?
Yeah. So what happens? So I see that it's almost always a foot, but sometimes it's a head, and also in the +1 defect, mostly a head, but sometimes a foot. So are these instances which are, I mean, do not fit the norm? Do they have anything special about them? I mean, the process, like suddenly, because of some fluctuations or I don't know, changes to, let's say it started as a +1 defect and then eventually it became a foot. Does it change to 2 plus half defects, or it can just generate a foot from a +1 defect? I can't. Why it's not like always happen?
So it's a great question, and I'd say a little more. So the examples I've shown you here are examples which are kind of the very nice examples where the defects are very clear. In general, even when you look at the same region over time, especially in the defect region, so the regions that are well ordered are well ordered. The defect region, there's some local disorder. It's not totally well defined. What's a +1 defect and what's 2 plus half defects. When the way we defined it here was having some distance between the plus half defects, but it's not super well defined. And I think I'll get into the this just now about. I mean, the nematics doesn't give us all the information. Obviously, there are other things in the tissue. What I think is clear that if I have a region that is like a +1 defect, that locally there will be a different environment. Is this how different if these 2 plus half defects are close? How different is it from the environment here? That's, I think, is a more. I mean, it's not a discrete difference. I'll put it this way.
Okay.
Any more questions, or should I keep going?
Thanks. I have just a curiosity. So these cells, when they remodel the tissue, they basically contract, or they do something else. I don't know, like they slide on each other. What is their main task?
So the dynamics. So if we look at cells, I don't have movies showing it here, but we've looked at the cells. If you look at the cells, you can see that they're definitely moving. So it's in epithelial. They're not mostly within the epithelium. Mostly you see the epithelia deform kind of in a continuous manner, but they do they can change neighbors, and eventually when the animal grows and elongates, you will see changes in the neighbor, but it's as little as needed to accommodate the changes in the shape. So there's not. I'll show some movies later when we track. I can show it now. In a couple of slides, I'll show, for example, movies where we labeled a larger region of cells. Sorry. A larger region of cells, and you can see that there's certainly deformation, and there's certainly some changes in shape. There's certainly neighbor exchange, but there's not much motility. I mean, the region remains kind of well-defined, connected region.
Okay, so we have a question from a remote participant. So it's Camilo Andino. He said, "Thanks for the nice talk so far. I was wondering if you can assign some sort of energy function to the defects and whether the emergence of the emerging annihilation of the defects would be favored or not."
Mm-hmm. So that's actually something that we're interested in looking. So for now, the description we have is totally phenomenological. If you think of our system kind of in the theory of active nematics, then it's a complicated system because we have it's deformable. So it's not sitting on a fixed geometry. which implies that if you think of some, if you think of some energy associated with a nematic, it would obviously depend on the local curvature, but the local curvature is changing, and it's changing in a coupled way to the nematic field, but the coupling is not simple because the fluctuations due to the contractions are much faster than the changes in the shape of the in the organization field. So I'd say that it's interesting, certainly interesting question to think if we can, how much we can basically, if we think of modeling the nematic field, so we have the field and we think of modeling how much of the dynamics that we see of the nematic field can we capture with such a model, which would have to somehow take into account both the nematic field, but also the deformable surface it's sitting on. Currently, we don't have any concrete model, but I think it's an interesting direction to pursue. And I'm sure that not everything will be explained by the nematic field, but it's an interesting question how much of the dynamics we can explain by just thinking of the nematic.
See one more question.
Hi, thank you. Do you think it would be doable to generate your own defects in the nematic field?
Mm-hmm. So that's an excellent question, and so one thing you can understand from the having a nematic field is that you cannot okay. So naturally, okay, let me start with naturally the system. What we see in the system is that there's the defects are not easy to generate. So if you look naturally on the system, early on in the regeneration process, we don't see any new defect formation. We just see defects form in the disordered region. We do see new defects forming during the emergence of the tentacles and during, if you look at a full animal, during budding. In these processes, what you see is that the defects do not emerge in the flat surface, but actually the formation of defects is associated with the formation of like you can see here, the defects form as you see a deformation. So we have a high curvature region, a bump appearing with the defects forming. So certainly it's possible to form defects in the system. In these cases of budding and tentacle formation, I would think that there's some pre-chemical and pre-signaling processes that initiate this formation of the bump and formation of the defects. Experimentally, it would be great if we could generate defects. One thing which is true both for the natural system and if we try to manipulate it is that if you want to generate defects, you cannot just generate a +1 defect because that will not conserve charge. So you'll have to generate, like in tentacle formation or budding, what you see is +½ associated with 2 -½ forming as is. We never see, for example, a formation of +½ and -½ pair of defects, which is the common type of excitation you see in other nematic systems. This we never see. What we see is always +1 and 2 -½ associated with a curved surface. What we are trying to do is apply localized forces, for example, and see if by that we can induce changes in shape and defect formation. And the other thing that we can do is play with initial conditions and have situations which are more, will lead to more defect formation than the minimal amount you need by the topological constraint. So it's a very interesting thing, but it's not trivial.
No more question. Thanks.
Okay, so one obvious question that arises from what I showed you is that, okay, so this +1 defect that eventually becomes the head of the animal, this is where the organizer developed. This is the main thing that is associated with the formation of pattern in the animal. Where is it located? So I showed you that we have initially in the spheroid, we have kind of half of the spheroid is organized, half of the spheroid is disordered, and the defect will appear in this disordered region. But where in this disordered region can we say something about where the +1 defect will form, or associated with where the new head of the animal will form, and what determines basically the polarity, the axis, the animal axis, the body axis of the regenerating animal. So to look into this question, and I guess another thing I want to emphasize, I said that if I sit on these fibers in this region where we have the inherited the order, I can sit on these fibers and I will eventually find myself aligned with the axis of the regenerated animal. These fibers will remain stable throughout the process, and eventually will line up with the body axis of the regenerated animal. But the fibers themselves are likely nematic. They're not polar, so the polarity does not come from the fibers themselves. So to look into this question, what we did is we wanted to label the tissue in the original excised tissue segment and see where the head forms relative to the original tissue organization. So we wanted to have to label both the head-facing edge of the tissue and the foot-facing edge of the tissue. In order to be able to label with two colors, what we did was generated grafted animals where each half was electrorated with a different cage dyes, and then when we uncage them, we can get different labels for the head side and the foot side. So you can see here we have the excised tissue, and we have labeling in magenta on one side and in green on the other side. When the tissue falls, as I mentioned earlier, this is not a trivial thing in terms of the organization of the fibers and also in terms of polarity. As you can see here, the foot-facing side and head-facing side of the tissue come together. Now we can follow the dynamics of the tissue. So again, we're starting from the folded spheroid here. You have the head-facing side labeled in magenta, the foot-facing side labeled in green, and you can see the dynamics. The dynamics of the tissue. So related to the question that was asked earlier about the tissue dynamics, so you can see here there's a lot of deformations in the tissue, but you can see that the labeled regions roughly remain pretty well defined over time. And what we were surprised to find is that, so you see the +1 defect forming in the magenta region, and this will become the head of the new animal. And this is invariably the case. So what we see is that invariably when we label the head-facing side of the tissue and the foot-facing side of the tissue, the +1 defect doesn't form randomly in the disordered region, but it always forms in the region that came from closer to the head, so the magenta labeled tissue. And also note that it doesn't come from the edge of the tissue. So it comes from cells that were closer to the head, but not the cells that were closest to the head, which are the cells that were at the boundary of this labeled region, but kind of a few cell distances into the labeled region. So here you can just see snapshots from the movie. So this is the initial spheroid, folded spheroid. You see that this is the large disordered region. So on the top, you see the actin fibers, and you just see the outline of the labeled region. Magenta is the head, what was originally in the head side. Green is what was originally in the foot-facing side. You see early on, so this is 11 hours after excision, well before any morphological features appear. We see a +1 defect form. Invariably, it forms within the region that came from the head side. And this region will remain, this defect will remain stationary relative to the underlying cell. And eventually, this will become the organizer of the new animal. So this is the mouth, the tip of the mouth where the organizer of the hydra head forms. So what we see here is that the defect side, on the one hand, there are constraints from the nematic that lead to the formation of the +1 defect in the disordered region. But on the other hand, there's also some polarity information, presumably some biochemical signaling processes associated with the head side that lead to the formation of the defect within the region that was closer to the head. And in general, this result was somewhat surprising to us because what we see is that the memory of polarity is very strong in the tissue. So even though the tissue folds in a very non-trivial manner, bringing the head side close to the foot side, when we look at the regenerated animal, we see that the head side is generally in the originally head region closer to the head is found at the head of the regenerated animal, and the region that was closer to the foot is found in the foot of the regenerated animal. Okay. So, so to summarize this first part, I've shown you that we can look at the organization of the supracellular actin fibers, and we can look at this process of regeneration from the viewpoint of this actin fiber organization and active nematics. And what this gives us is a coarse-grained framework to look at the process of regeneration. We can identify that there are defects in the nematic fields. We can identify them early on in this process. And we can see that these defects act as organization centers. So these will be the sites where all the action is happening. Now, that said, these are certainly not the only processes involved. And this is intimately coupled to other processes. And this is highlighted in these movies where we see that the polarity of the tissue is preserved in this complicated way, despite the folding process that brings the head side to the foot side. So if we just look at the nematic field early on, the tissue is symmetric with respect to the head side and the foot side. We don't see any differences, particular differences in the nematic field early on. Over time, what we see is that the +1 defect always appears in the originally head-facing side. The 2+½ defects will form in the other region and then will eventually become the foot. So it's a really nice example where you have both the mechanical information in the nematic field, but also some additional information, presumably from biochemical signaling pathways that together help shape the animal. Any questions?
No questions, thanks.
Okay. So let me move to the second part of the talk where I focus on the polarity of the regenerated animal. So, okay, so we know that the actin fibers give us a memory of the alignment of the tissue. I said that fibers themselves are nematic, so we have the alignment but not the polarity of the tissue. When we look on conditions where we have clear-cut initial polarity, this polarity is well preserved, and this has been well known in Hydra for a long while. So from the original experiments by Tremblay, it was known that if you take half animals, they will regrow according to the original polarity. It's been known for many decades that if you take a ring of tissue, this ring of tissue will preserve its polarity. So you can see an example of that here, where we put the ring of tissue on a wire just to keep track of its orientation. And you can see that the head side will always form the head and the foot side will form the hook. And even more surprising is what I showed you just now, that if we take a piece of tissue which folds in this non-trivial way, still polarity is preserved, and we see the head side being primarily at the head of the regenerated animal, and the foot-facing edge primarily at the foot of the regenerated animal. So there's a very strong memory of polarity in the tissue. And what we wanted to look into what determines the polarity of the animal, what we wanted to do is generate frustrating conditions. So what we wanted to do is generate initial conditions where this memory cannot be simply, we don't have a simple memory of the polarity. And the way we generated this frustration is by taking two rings and fusing them to each other. So the idea is that we take two Hydra, we cut off rings that have very well-defined polarity. Each of them have a very well-defined polarity, but then we connect them into this doublet tissue. So we adhere the two rings to each other. What we get is something that looks like a thicker ring, so it will seal in the top and bottom, but the middle part will connect between the two rings. And then we can play around with the polarity and the position of these two rings in the fused doublets. And what you're seeing here, this is what we call the head-to-head configuration. So we take the two rings and we put them attach them through their head-facing side. And the question, how will the system reorganize its polarity under these frustrating conditions where the upper piece has a polarity in one way and the bottom piece has a polarity in the other way? And we can use this perturbation to try to understand what's important for determining and establishing the polarity during regeneration. So when we take these two pieces of tissue and you fuse them together in this head-to-head configuration, the naive outcome, if you think that polarity is strongly preserved, would be to have a head in the middle. And indeed, if we look at a large amount of samples, this is what we see. So what you can see here, these are, we have the two different rings. They're fluorescently labeled with different colors. You can see that initially it looks like a long tube, but over time you see a bump forming on the side, and you can see a new head forming in the middle of the tissue. Okay, so this is kind of what you naively would expect if the system maintains its polarity. You get a head in the middle. But what we also see is that in a large fraction of samples, this is not the case. What we see is that the system actually heals smoothly, and we actually develop a head on an originally foot-facing side. So here you can see in this movie we have again the same initial, nominally the same initial conditions. In this case, the interface remains smooth. There's no morphological features developing there, and we see a head forming. And it doesn't form anywhere in the tissue. It forms on the edge of the foot-facing side of the tissue. So what we see here is that this polarity, which has a very strong memory, if we put it under frustration, it actually can be erased. And moreover, we see that there's some bias towards the edge of the tissue where the new head forms, even though from a kind of signaling point of view, this would be the least likely place to have a new head. We can play around also with not only with the orientation, but also with the relative position of the two tissues. So here what you see is what we call the head-to-foot configuration. So here we take two rings and we connect them with the same polarity, but we can take the rings from different regions of the animal. So we can take the ring from the upper part or the lower part relative to the mid-body of the animal, and then we can connect them as they were in the original animal, so with the upper part above the lower part. So this would be the same as in the original animal, or we can flip them in what we call the anti-oriented direction. So we put the lower body part above the upper body part. What we see is that if we do it in the kind of, if we keep the orientation as it was in the original animal, invariably we get normal animal. We get a single head at the head-facing side, no head at the interface. So again, the individual rings would generate a head in the interface. But when we put them adjacent to another tissue, this is inhibited, and we get a single head along the same polarity of the original animal. When we look at the anti-oriented configuration, actually we have a miss. So they're very different morphological outcome. We have some of the animals that regenerate along the original polarity, but we have a lot of other things. We have heads growing in the middle, and most striking, we have animal that flips the polarity of both rings. So we have an animal that has both rings going the other way, and a head forming, as I showed you before, on the foot-facing side. You can see an example of that here. So again, we are in the head-to-foot configuration, but we put the lower part of the animal above the upper part of the animal. Initially, again, we have this cylindrical shape with the two edges, and you see that the head is forming, and it's forming on the foot-facing edge on the opposite side of the tissue. So this is quite striking. We have the polarity flip, and again, we see this bias towards the edge. The head doesn't form anywhere, but it forms at the edge of the tissue. So kind of to summarize this, we see that when we put the system, if we initially took the system, the head side will always develop a head, a ring, and a foot will always develop a foot. But now if we put the system in frustrating conditions, we see that we can observe head formation on an originally foot-facing edge. So again, the region that is furthest from the head in the parent animal will develop a head. We see it both in the head-to-head configuration and in the head-to-foot configuration when we flip the positions of the two pieces. And we also see that there's suppression of head in the middle. So when we put when we attach the tissues in the middle, we have, whereas in an individual ring, all the sides would form a head. When we put the head side adjacent to another, we're likely to suppress the formation of another head. Okay, so this was just phenological description without going into the fibers, but let me now relate this to what's happening with the actin fiber organization. So when we take individual rings, each ring will have this parallel array of actin fibers. When we attach them to each other, What will happen at the middle interface, if we do this cleanly enough, what we'll see is that the actin fibers can connect and form this seamless connection between the two parts. And at the edges of the tissue, we'll have it sealed into a +1 defect as we see in an individual ring. So if you look at the actin structure of this ring doublet, basically it looks like a thicker ring with 2 +1 defects at both ends of the rings. Now, when we look at the polarity reversal, so this is this example where we had head-to-head configuration. We have suppression of head formation in the middle. We have the head forming at the edge of the tissue. If we look at the actin fibers, so here you're seeing snapshots from a time-lapse movie. The upper parts are just snapshots where the tissue happened to be on the side, so we can see a nice side view of the tissue, and the bottom are the same tissue, but just frames where we see the top view of the image. What you can see, you can clearly see this +1 defect that forms immediately as the ring seals, so you can see this +1 defect at the edge. You see the nice interface in the middle with no defect formation, and you can see that when a head forms on the foot, initially foot-facing side, it will form exactly at this +1 defect. So what we see is that we have this, from the structure, we have a +1 defect on the edge, and the site of the new organizer formation, new head formation, is the site of this defect. Now, as I mentioned earlier, this is not the only possible outcome, morphological outcome in this experiment. In other cases where we see head formation in the middle, what we see is de novo defect formation. So initially, this region does not have any defects. But then what we see is that the formation of a head is associated with defect formation that eventually will lead to the formation of a head here. And again, this is smack at the interface. So what this is telling, so it's again a nice example showing you the interplay between signaling and the nematic structure. So on the one hand, if you introduce this frustration and there's strong signaling that wants to form a head in this region that lacks a defect, well, it lacks a defect, so often we'll have suppression of head formation, but this still can lead to head formation through the formation of new defects. But if this suppression is strong enough, then the system will look for alternate sites for forming a head, and the defect will actually act as an attractor, be an attractive point to generate this, head formation. So to summarize this second part, I showed you this ring doublet system, which is a nice system to introduce frustration in polarity and look at the how polarity is reorganized in the system. It's clear from our movies that polarity, even though there is a strong memory of polarity, it's actually, this memory is not irreversible. You can actually erase it and even reverse it. We see that this polarity depends on an interplay of different factors. It depends on both the original tissue polarity, its position along the body axis, as well as the structure, which is associated also with the actin structure, that having or not having a defect. And we see that the +1 defects can act as attractors for head formation. And now I say this, it's a complicated process. It's not that the +1 defect defines having a head. This is obviously not true. If you look at a single ring or a ring doublet, we have +1 defects at both sides. One will become a head, one will not. We also have +1 defects at the tips of the tentacles. So certainly there's not a direct association between having a +1 defect and having a head, yet all the heads will always have a +1 defect, and it seems that having a +1 defect can direct the processes to a particular spot in the tissue where the defect is localized. Okay, so to kind of wrap up, I've shown you that if by looking at this supracellular actin fiber organization, we have a mechanical field, the orientation field associated with this actin fiber orientation. And just by looking at this field of this mechanical field and the defects it contains, provide us useful information on the regeneration process. And in particular, it's predictive. It's predictive in the sense that we can identify regions and know that these regions, what will be the outcome of these regions at the end of the morphogenesis process, just by looking at the nematic actin fiber organization. So we have some effective description of which defects arise, what will be their dynamics, and how they will associate that with the regions that develop into the morphological features in the animal. But obviously, this nematic field is not all the story, and we see both when we look at the position of the +1 defects in the strips, and we see that it's always in this originally head-facing side of the tissue, or in these examples where we look at the ring doublets, and we see that the relation between the polarity, the morphological outcome, and the original polarity and structure in the tissue, we see that there is additional input that goes into determining the polarity in the tissue. And what this highlights is that these nematic field and these signaling processes need to be tightly intertwined to generate these very robust outcomes in the animal. And what this brings me is to the idea of a mechanical morphogen. So the kind of dominant view in development is based on chemical morphogens. So the idea is that there are these signaling molecules, as I mentioned in the beginning of my talk, and they're concentrations of so they're concentration fields of these, which can vary in space and time. So they can have reactions, you can have transport through diffusion or through other means. And the idea is that these provide local information. So these concentration gradients provide local information to the cells in the tissue. The idea I want to put forward is today is that we can think of a mechanical analog of this. We can think of mechanical morphogens. So if I think of the orientation field of the actin fibers, I have a field that varies in space and time, and it integrates information from the mechanics of the tissue as well as from biochemical information into this, which will influence its dynamics. And this mechanical field also provides local information to cells. So if I look at the local defect environment, which are singularities in this field, they will provide a unique mechanical environment. And this mechanical environment can push the system towards developing a head organizer in the site. So the idea is that we can think of these mechanical fields as mechanical morphogens. Obviously, they're not acting on their own. They're interacting with other mechanical and biochemical morphogens, and eventually the feedback between these different fields is what leads to the morphogenesis process and the formation of functional tissues. So with that, I'd just like to thank my co-workers on this project. The work I showed today was the work of Yonit, Lital, and Anton, who are graduate students in our lab, as well as Lior Garion, who's a research associated. And all this work is done in close collaboration with Professor Erez Braun, who's my colleague in the physics department. And I also mentioned that we have open positions for postdocs or graduate students. If anyone's interested, I encourage you to email me so we can discuss this. And thanks, and I'm happy to take more questions.
So we have a question from a remote attendee, Camilo Andino. So I said, thanks again for this really amazing talk. I was wondering, when you cut out a piece of the tissue and it regenerates, it must also regenerate the different cell types, muscle, neurons. Do you know about the temporal and spatial correlations of the nematic fields and the regeneration of the different cell types?
Okay, so I'll start with saying that for the regeneration per se, you can actually do that without any neuronal or other types of cells. So if you just make hydra that are just with ectoderm cells and endoderm cells, so you have just the endoderm and ectoderm stem cells, which basically the bulk of the tissue in hydra contains these stem cells. They get differentiated only in the extremities of the animal. You can get the regeneration. So our focus has really been on the ectoderm and endoderm cells, but obviously the other cell types, in particular neuronal cells, are also involved. The migration, for example, the migration of other types of cells is faster than the migration of the epithelial cells. We haven't looked at all at the differentiation patterns in other cell types, but I'm sure they are correlated with with what's happening with the epithelium.
So thanks again for the nice talk. So I have a question. Probably you mentioned, I missed it, sorry. When you attach and connect two different tissues, what stops them from making two individual and separate animals?
I mean, there's nothing stopping them, but if you put the tissue in proximity with another tissue, this does not happen. So the tissue is able to, I mean, if you wait long enough, the tissue will seal on itself and you'll get to animals. But if you attach them quickly after excision, they will attach to each other and then they will form basically a single tissue.
So what kind of bond we are dealing with here? I mean, between the two tissues?
So basically, you have cell-cell adhesion, cadherin associated cell-cell adhesion that is generating this cell-cell adhesion as it is within the tissue. I should also emphasize that there's a strong wound response. I mean, once you excise the tissue, this has been seen by many people now looking at the molecular, various molecular components. There's a strong injury response when you do that, but when you put another tissue close by, it will seal into a single epithelium and generate. I mean, you can sometimes get, as I showed you, multi-hooded animals, but typically the tissue will remain as one connected component.
Okay, thank you.
Hi, it was really amazing talk, so thank you a lot for that. I have two questions. The first one mainly about the same experiment of the grafting, but if you've done it with your blender experiment, so like with dissociated cells, to see if it's something more intrinsic with the cells or is it like a more tissue level property that decides polarity. And the second one, in regeneration experiments, did you try putting blebistatin, and if that affected at all how the defects moved and affected polarity later?
So first, regarding the first question, so people have done, we haven't done these experiments with spheroid. People have done some experiments where they took cells from the organizer region in Hydra and mixed them with cells from the general population. And what you saw is that, what they saw is that indeed clusters of, I think, very small number of cells from the organizer region were sufficient to bias the formation of new head structures. So certainly, if you have a small number of cells from the organizer region, can direct the the morphogen. Regarding if I think so. So in principle, all the cells, all the regions of the Hydra are capable of forming a new organizer. So if you took all the cells in the but they have to form it. So if you take the organizer itself, it already has these features, where all the cells in the rest of the tissue can develop into an organizer, but not an organizer to begin with. So if we took just regions from the mid part of the tissue and blended them, I doubt that there would be a strong association between the head region and cells of a particular type. Regarding your second question on blebistatin certainly influences the nematics dynamics and the organization process. So if you use large amounts of blebistatin, you totally block regeneration, so you don't see any regeneration. But this takes a few hours. If you look at the nematic actin field, you actually see that it's totally disrupted. So when you put a high concentration of blebistatin, within a couple of hours, you'll see the nematic field destroyed, and within a few more hours, the tissue will break up and die.
Thank you.
And so hi, thank you for the nice talk. And I wanted to ask like a very general thing. I'm not sure if I understood it. So you said that these defects are arising from folding, right? So what makes you think that the mechanical morphogen is the actin fibers instead of the, if the actin fibers are organized in a certain way as a consequence of folding, then why is the mechanical morphogen are the actin fibers instead of the tissue curvature itself?
Okay, so let me start by maybe explaining what I mean that the defects are formed by folding. What I meant is that when the tissue folds, you change the topology of the system. So you start with a flat piece of tissue with open boundary conditions, and then you close into a spheroid. When you take the region initially has open boundaries and no defects. It's parallel everywhere. Once you close it into a sphere, it cannot be parallel anywhere. You cannot have something that's parallel everywhere without any defects. So there must be, if the system maintains its nematic order everywhere, there must be defects. So this is what I mean. So it's not that if I have a nematic on a spheroid, so if I have a hydra that closes into a closed ball of tissue, a hollow ball of tissue, then I know there have to be, there's nematic order everywhere, so there will be localized nematic defects. I know there must be defects. If I look at the local curvature, or for example, if I look at the shape of cells, this will be highly fluctuating. You saw the movies of the tissue. The tissue it's basically like a muscle. It's contracting and changing its shape and deforming very on all time scales. So you have very, if you look at tissue curvature, or if you look at the shape of cells, these will be highly fluctuating over time. There will not be any kind of well-defined pattern. When you look at the nematic field, we very clearly see, you saw in the movies, you can follow this nematic field. The orientation is actually quite stable despite all these deformations.
Okay, one more question from Camilo. So thanks for your answer. A related question. Does Hydra have any immune response that is triggered when cutting out pieces of tissues?
Yeah, certainly. So there's definitely a strong injury response. It's been seen it's been studied recently by several labs, and definitely once you cut the tissue, there's a strong signaling response. Not all the details are clear, but it's definitely that many factors are triggered by this by the injury.
I think we're done with questions. Thank you for all of your answers.
Okay, thank you very much. Enjoy the rest of the session.